So I'd like to introduce our speakers today. This is Dr. Pam and Doug Soltis. They're both distinguished professors at the University of Florida's Natural History Museum. And during their work, they use molecular data, herbarium collections, and computational methods to study plant diversity at multiple phylogenetic and geographic scales. And although their work focuses on the plant branch of the tree of life and the evolutionary processes that generate diversity, their research also has implications for conservation and the management of plant diversity, particularly in Florida, both in the present and under models of climate change. So Doug and Pam, thank you very much for sharing your time with us, and I'll let you take over.
Thanks a lot, Lisa. It's really a pleasure to be with all of you. And it's great. Whoops, let's see here. It's great to be able to share. Okay, am I unmuted again now?
Yes.
Okay, great. It muted me for a minute there. So I'll just back up again and say thank you very much for the invitation, Lisa, to be able to present to the audience today. It's really great for Doug and me to be able to spend time with a bunch of Florida plant enthusiasts. So we're looking forward to having a great discussion with you after our presentation. Also, Doug and I will be presenting this talk as a tag team. So I'll start with the beginning and give our introduction and some of the methods, and then I'll pose a question, and then I'll turn it over to Doug to let him do the answering. And then we'll both be available for question and answer time at the end. So as Lisa mentioned, we are interested in plant diversity. We focus some of our research on the big picture of how plants are related to each other, but we also use a variety of techniques for trying to understand better how rare species may respond to climate change and how we might be able to use the tools of ecology and evolutionary biology to help with conservation efforts. So species are not distributed randomly or evenly around the globe, but instead they're clustered based on various types of resources. And these clusterings of species are referred to as biodiversity hotspots. Now, until recently, there were 35 global biodiversity hotspots, but recently, within the past 6 or 7 years, a new biodiversity hotspot was recognized, and that's the North American coastal plain. And that is indicated there by the yellow arrow. So what is a biodiversity hotspot? Well, first of all, it is an area that has more than 1,500 endemic plant species. But it's also an area that has undergone extensive habitat loss, more than 70%. So this combination of high species diversity and high habitat loss means that these regions are conservation priorities. And again, the North American coastal plain was recognized as a biodiversity hotspot in 2015, and it provides a lot of really valuable information for understanding biodiversity hotspots in general. And part of the reason for this is that this is an area that has been under settlement for a long period of time, and yet it still retains a lot of biodiversity. So just to orient you a little bit, this biodiversity hotspot runs from essentially the mid-Atlantic region down along the southeastern coast. It encompasses most of Florida. Then it goes along the Gulf Coast all the way to eastern Texas. And then because of the low elevations around the Mississippi River, it actually goes quite far north through Louisiana and northward there. Now, although this is a fairly large region and it has approximately 6,000 plant species, this biodiversity hotspot is a little bit bigger than we want to focus on for our discussion today. And so we're going to be focusing just on Florida. But before we start talking about the plants of Florida, we'd like to start out with some key points. And these are some of the main points that we'd like to make during our presentation today. So just in case you need that after lunch nap and you doze off a little bit, here are some of the main points. So first of all, the Earth's climate is changing. That's not up for debate. So in order to cope with that, species have three options. They can either move to areas where the habitat and the environment is still suitable. They can adapt to the new conditions they find themselves in, or they can die. And this has been referred to as MAD responses: move, adapt, or die. Now we can expect a number of changes to our Florida plant communities over the next 30 to 50 to 100 years. It's been predicted that many, many species on Earth will be extinct by the turn of the century if things aren't, if we don't change how we interact with our environment. And so there's incredible impetus for us to understand the species we have today and to then consider ways to conserve them. Fortunately, we have a number of new tools that can help us predict future distributions of plants and other organisms, and this can then help us develop conservation approaches. So family trees of plant relationships can help us assess areas for conservation. And this, although this idea was introduced about 30 years ago into the scientific literature, it's something that is still sort of a novelty when it comes to applied conservation. And Doug will be talking about this more a little bit later. And finally, what I'd like to mention is this idea that what's old is new. Herbarium specimens, some of which may be centuries old, can provide the data for helping us to understand the future distributions of species and what we might be able to do to help in their conservation. Now, some of you may be familiar with herbarium specimens if you happen to have taken a taxonomy course along the way. And people sometimes think of these as, you know, being important tools, but they're tools for taxonomy. What we know now is that these herbarium specimens provide us with a ton of data that can be used for conservation, understanding ecology, understanding evolution, and more. So let's focus on the flora of Florida. So the flora of Florida has about 4,800 species of land plants, 3,300 of which are natives, leaving about 1,500 being non-natives. And of these, there are about 4,300 species of vascular plants. And again, about 3,000 native, 1,300 non-natives. And those non-natives come in a variety of forms and dangers. And that would be a great talk to think about the effects of these non-native species, but that will have to wait for another time. Now, the flora of Florida is at risk. 600 species nearly are either federally or state endangered or threatened. And this just summarizes some of those. But I think the big thing to think about is that we have close to 600 species that are threatened or endangered. Now, our rare species and our threatened and endangered species sometimes occur in a very scattered way across the state, but sometimes they are found in very specific habitats or areas, such as the Apalachicola Bay region and also the Lake Wales Ridge. And so I'm showing here the Lake Wales Ridge as an example. There are many, many rare species that occur in this unique habitat, and many of them are endangered and threatened. And in fact, the Lake Wales Ridge National Wildlife Refuge was the first refuge that was actually designated for the recovery of endangered and threatened plants, because in fact, it's 35 plant species that are listed in that region and only 23 animals. I say only, but of course, this is quite a small region. And the fact that there are this many endangered or threatened species is very, very serious. And this is also an area that has only about 15% of its native habitat still intact. Now, the habitat loss in the Lake Wales Ridge has been due to many factors, such as agriculture, golf courses, housing developments, And the effect of all of these changes in land use has been suppression of the fires that were crucial for the maintenance and stability of natural ecosystems. And in some cases, even for the plants to be able to complete their life cycles. There are many, many plant endemics, just a few of which are shown here, and many other rare endemics as well. Some classic species such as the Florida scrub jay, the Florida mouse, which is the only endemic mammal to Florida, and even this amazing iridescent bee, which is called the blue calamintha bee. All of those occur on the Lake Wales Ridge, and again, many others. So it turns out if we could understand something perhaps about the plants of a region and we could develop appropriate conservation strategies, it makes sense then that we could conserve not only the plants, but also the animals and fungi and microbes that are also native to that region. Now, one of the ways that we know anything at all about where organisms are distributed is through our Natural History Museum collections. And there are about 1,600 such collections in the US, and they house somewhere between 1 and 2 billion specimens, and somewhere between 3 and 4 billion specimens are in natural history museums worldwide. And these are some amazing photographs from the Smithsonian. With regard to herbaria, those museum collections that house plants, there are about 3,400 herbaria worldwide, and they house about 400 million specimens. So there's just an immense amount of data about species diversity and distributions in our herbaria and more broadly in our natural history collections. But as I alluded to earlier, typically we think of these collections as being the purview of systematics and taxonomy. And of course, they do form the foundation for all of our nomenclature and for most of our understanding of species limits and species distributions. But in fact, these collections can do much more. They can provide us with keys as to the genetics of the species. We can even get whole genomes from museum specimens now. We can study chemistry, species interactions, phenology, that is the changes in the life cycle of an organism over seasons, biogeography, and many, many more topics. The problem has been until the last maybe 15 years or so, that most specimens in natural history collections were locked away in cabinets and they were unavailable for general use. But through new technologies and new funding from the National Science Foundation in particular, digitization is becoming much more widespread. And this means that information about the specimens and sometimes images of the specimens themselves are being made increasingly available online. So for a plant specimen, digitization might be capturing the information on the label. And typically on a herbarium specimen, this is a beautiful specimen of a pitcher plant from Calhoun County. The label information is usually found in the lower right-hand corner of the specimen, and it contains things like the scientific name, the date of collection, the collector's name, where the specimen was collected, including GPS coordinates if those are available. It might also list something about the species that are associated with it, and there might be some other additional notes. Now, if all of that information for every one of those several, well, maybe 400 million plant specimens were captured and available online, we would, as scientists and conservationists and the public, have access to really vast amounts of information that can be put to all sorts of uses. Now, that process is ongoing, but it is indeed a little bit slow. But in addition to that sort of label information, there is also a lot of imaging that's going on. And this includes images of herbarium specimens, like you can see on the right, but also various types of imaging of other organisms as well, including sometimes 3D imagery that allows one to even investigate the internal structure of an organism. Now, all of this information is being compiled into a huge database called iDigBio. And this is a project that is a collaboration between some of us at the University of Florida and some colleagues at FSU. And so we have a lot of great collaboration going on here, despite what our athletic teams might be doing. So this is the National Coordinating Center for Digitization of Biodiversity Collections. And it's at this point a 10-year project funded by the National Science Foundation, but we are hoping that there will be funding to enable the center to continue its operations because of its importance to the whole biological community. So this database serves information at the moment for almost 127 million specimens. So, and these include plants, fungi, animals, various types of algae, lots of diversity here. And there are nearly 41 million media records. Now, most of these media records are photographs, and most of those photographs are herbarium specimens. So if you're a plant biologist, you're in really good shape because there is so much information available through the collective actions of our herbaria. Now, if we look at a heat map of specimen distributions in iDigBio, we see very dense coverage in the US, but also throughout the Americas and throughout other parts of the world as well. So the dark red to almost brown colors indicate a high diversity of species. And you can even tell that there's the density of specimen records is very high in Florida because Florida has been very effective at doing digitization. Many of our nearly all of our collections statewide have some data available in iDigBio. So you can search this database, and I would invite you to do so. I put the URL on here a few times. You can go to idigbio.org and go to the place where it says search the portal, and you can search for any species using the scientific name. So here I've entered under scientific name Acer rubrum. This is the red maple. And this map then will return the points for every specimen that has a geographic location associated with it. And so you can see this very dense collection of points in the eastern US, which is of course where this species occurs. And we can look at this a little bit more closely, and you can see that there are a lot of dots on top of each other showing very, very dense sampling. It's also possible to hone in on a specific point and pull up the specimen record for that point. And then that will also tell you a little bit about it, the species name, the family, splits it into the genus and species. And it also tells you whether or not there are other data available, such as a photograph. And then you can click on that and get a very high resolution image. And this image can then be studied for aspects of morphology. One could score the flowering date and then compare that across time to see if there have been changes in flowering date with climate change and so on. And again, lots of information about plants. Here is the pie chart of the specimen records. 47.6% of all this 127 million specimen records are plants, and 85% of the media records are combination of images and recordings, audio recordings are for plants. Now, iDigBio is only one of these sorts of what we call aggregators that's available worldwide. There are some other aggregators or portals that are specific to their countries, such as the National Specimen Information Infrastructure in China, Canadensis in Canada, the Atlas of Living Australia, Species Link from Brazil, and so on. And then there are other ones that are focused on specific organisms like VertNet, which is for all vertebrates. And then there's a global aggregator that sort of aggregates the data from the other aggregators, and that's the Global Biodiversity Information Facility. And as a global facility, it's funded by a consortium of international partners, and the National Science Foundation contributes to its operation as well. So let's return to biodiversity hotspots. We have hotspots scattered across the US, and this shows what these hotspots look like, not in the realm, across the realm of the whole globe, but just with regard to the US. And we can see that there are several such hotspots in Florida, others in the Appalachians, a number of them in California, and certainly in Hawaii, and a few others spread around. So given the distributions of these hotspots, given what we know about different sorts of ecosystems, what do we protect? How do we make the decision as to where our conservation efforts and our conservation funding should go? And rather than me try to answer that question, I'm going to pass that difficult question on to Doug and let him describe some of the efforts that we think are important ways to go about addressing this important question.
Okay, well, thanks, Pam. And as Pam noted, we're really happy to be here. So I'll go ahead and take us through the next part of our presentation. So what we're going to do is focus a little bit on Florida plant diversity. We owe a lot of this work to great collaborators such as Julie Allen on the right, and then Rob Gralnick on the left. Rob is a faculty member in the Florida Museum, and Julie was a postdoc, but is now at the University of Nevada. So what I'm going to do is introduce a new concept, and it's a big word, but hey, it makes a lot of sense. We'll explain it. And that is the concept of phylogenetic diversity. What we're really talking about when you talk about a phylogeny, it's just a tree of relationships. And so you're asking a really basic but fundamentally important question, and that is, how much of the tree of life is present in a geographic area? That's what we mean by phylogenetic diversity. So let's just have a very simple example. We have two areas, one on the top, one on the bottom. They have the same basic number of species, but the area on the top, these are just simple air models, are all oaks. That's location one. Now, some of those may be really, really rare. Okay. And so you think, well, we got to protect those. But on the bottom, we have all of these different highly diverse species that cover much more of the tree of life. So the question is, which one of these areas, location one or location two, encompasses more of the tree of life? This is not a trick question. And of course, if I could see everybody, we could have a raise of hands. And the answer, of course, is location two, because of the great diversity of species there. Now, that doesn't mean the area on the top with the rare species might not be important to protect, but the area on the bottom encompasses a lot more of the tree of life, has higher phylogenetic diversity. And that's what we want to talk about now. We all know that trees matter. Trees of relationships are important. Think of your own family tree. There you are, say on the bottom right, and you know, based on your relationships, your ancestors, if there's a certain disease that's inherited, well, there's a certain probability that you inherited that trait. Trees matter, relationships matter, and that's true for the tree of life as well. So trees of relationships are really important today in the study of biodiversity. And so what we did with our collaborators is that we built a tree, a family tree of relationships for the plants of Florida. Really, we concentrated on the vascular plants. If you look at this tree, there are ferns in there, gymnosperms. Most of this is flowering plants. That's mainly what we have in Florida. And we continue to add to this so that we can really encompass most of the 4,000 species into our tree for Florida plants. Now, Pam has already told you about locations. We have all of these great locations for plants in the state of Florida through repositories such as iDigBio. So now what we want to do is combine those. Okay, so here's our map of Florida on the lower left. We're going to divide that into a bunch of, they're pixels, little red dots. I'm just showing you one of those on the map of Florida. But we've divided it into 8,045 little communities. And then what we want to do is ask for each one of those little red dots, how much of the tree of life occurs in that region. And so there's our tree again over there on the right. It may be hard to see, but for that little red dot, every species that's in that area is marked in red on this tree. And what we can now do is sum across all of those little pixels, all of those 8,045 pixels, across the entire state of Florida and get an idea of where is high phylogenetic diversity, where is low phylogenetic diversity. That is, what areas have the most tree of life encapsulation versus low tree of life encapsulation. And ta-da, drum roll, here it is. This is what phylogenetic diversity looks like for Florida plants. The areas that are darker green on this map are the areas of high phylogenetic diversity. The areas that are lighter and lighter in color have lower and lower phylogenetic diversity. So this is the first attempt, the first effort ever to do this for any group of organisms in our state. And other areas of the country and the world are doing this as well. So we can then ask, since we are real interested, all of us, in conservation, what areas are protected in Florida already, and how well do they capture what we now have learned about phylogenetic diversity? So on the right, we see a map of Florida, and in green, we see areas that have some sort of protection, some sort of conservation status. Let's just see how that compares with our areas of high phylogenetic diversity. And here they are, just some areas circled. In red, we see that areas that are protected, but they have low phylogenetic diversity. And then other areas in the blue circle are not preserved, but they really are very little preservation, but they have high phylogenetic diversity. So let's stress right away, this does not mean that we are not interested in protecting those areas that are low in phylogenetic diversity. Of course, every one of those areas is important for various reasons to protect. But a goal moving forward might be, could be, should be, let's protect more in these areas that have high phylogenetic diversity. It gives us a chance to protect, to save more of the tree of life within our state. And maybe that's another way to look at conservation priorities moving forward. They're not mutually exclusive. Of course, rare species are incredibly important, but areas that have high phylogenetic diversity, they're sort of the arc of botanical life. Let's protect some of those as well. So you might ask, how does this compare to where we as Floridians live? And this is kind of an interesting telling map. The different colors indicate, well, if it's blue, it's low phylogenetic diversity, but low population density. Green is high phylogenetic diversity and low population density. And then we have orange, low phylogenetic diversity, lots of people. And then red is high phylogenetic diversity and high population density. You can see that some of our major cities, like Tampa, Orlando, are actually centered in areas that have lots of people, lots of phylogenetic diversity. Moving forward, some of those green areas could be of special interest for protection, for conservation, because they have a lot of phylogenetic diversity and low population density. Okay, now Pam also said we want to talk about what Florida plants do, what might they do in response to a changing climate. Because Florida is really going to be, already is in many ways, ground zero for climate change, not only in change of the climate, but sea level rise. And we're surrounded by water everywhere except on the north and a bit on the west. So we've got a lot of special concerns here. So Pam introduced this idea, well, plants might move, they might adapt, they might die. Can we predict or project what a plant might do? And the answer to that is, in many cases, I think we're getting very close to being able to do that. And what I'm going to do is give you an example of how we can do that using a tree, okay? A phylogeny, a tree of life, tree of relationships. So on the right, all those little branches there, that's a tree. It's for one plant group, a relatively small group, maybe close to 1,800 species. It's had a evolutionary history of about 100 million years. It's been around a while. These are all flowering plants. It probably started out with a sort of an intermediate temperature optimum. And what we've done is, it doesn't matter what the species are. You actually probably know some of the plants in this little group. There are things like peonies in here, sweet gum is in here, stone crops, gooseberries, saxifrages. So what happened then is we just look at the colors. You can see that this group of organisms, this group of plants, spawns some little groups that are very cold adapted and some that are yellow and red, and they're hot adapted. The blue ones are cold adapted. Now just look at the colors. This is like a rainbow. Notice that if plants evolved to be cold adapted, they've been in that state for maybe 15, 20, 25 million years. They haven't changed. They have not suddenly spawned new species that are hot adapted. These kinds of plots using a phylogeny, we know that trees of relationships are predictive. These are telling us that these organisms are not suddenly going to adapt to a hot environment in the next 15, 20 years. They're locked in to certain habitat types. They're not going to change. And we can now do that for much of the flora, not only of our state, but really for much of North America and other parts of the world. This group, by the way, contains some of the most cold adapted plants. You can see this is a saxifrage up here in the northern part of Norway, as well as very dry adapted plants, such as this stone crop relative down here on the bottom. So again, the point is that these plants are not going to adapt. In many cases, plants are simply, well, they're going to have to move or they're probably going to die. And let's go and take a look at our state and see what kind of projections we might be able to make. So what we're doing now is, again, using collection data that Pam mentioned, is model the distribution of species. We can get all of that great information on location, and then we can look at things like environmental features, temperature, precipitation, soil. These are all available. At the bottom, you see all these little maps, and they're actually showing us the distribution of things like soil type and temperature. And then we can model the range of each species in our state, and then we can make projections on the future climate models as to where these plants could potentially live in the future. So for Florida, we've done this so far for about 1,500 plants, but we're aiming to do it for all of the species on our state. And you can see we've used about 500,000 georeference points. So let's just see what we got. Now, there are going to be winners and losers in a climate change scenario. Here's on the top is the flat spike sedge. The hot colors indicate, based on our models, where these plants could do well now. And you can see that's mainly in the very southern part of Florida, the southeastern part. If we jump to 2050, now notice it's almost all blue or light blue. So there's really very little area where this species is projected to be able to survive in 2050. Now, on the other hand, we have plants like the scrub plum, which is now pretty much limited in terms of its model distribution to the Lake Wales region. But now in 2050, you could see that it could potentially have a much wider distribution. This does not mean that it's going to suddenly appear in Jacksonville. It just means that it could potentially do that. It could potentially live there based on these climate models and projections. So let's see, whoops, next slide. Now here's another Lake Wales Ridge species that you can see also on the left is its present projected or possible distribution. On the right, future. It could do very well based on a climate scenario, with Florida having a much hotter, drier climate. But on the other hand, we have plants that are wetland plants, and we can see that the cumulative change in suitable habitat for our wetland communities in Florida is really thought to be severely impacted by climate change models by 2070. The red indicates areas of severe impact, and you can see that's much of the coastal and areas of our state, as well as much of the interior. So those species, when we do modeling, are severely impacted. So let's summarize. We've been looking at thousands of plants now. Here's on the left are areas where what we have today in Florida, areas of high species diversity in green, lower as you get into yellow and tan, and then what we were projecting for the future in Florida. And what you can see is there's a lot less green, a lot more tan. As you might expect, many species in Florida, because we have a very temperate flora to our north, are projected, if they can, to move north, to migrate out of our state. There's actually some projections from our models of some species moving a bit to the south for areas where there may be, on projections, more moisture. But the bottom line is Florida is going to be a much less diverse state in the future in its flora based on these projections than it is at present. Now, that doesn't even incorporate sea level rise. If we had sea level rise, you can see that Florida is going to be a very changed state. This is one of the higher models of sea level rise, 4 meters. But regardless, that is also another scenario that we're trying to incorporate into these projections. So let's conclude. We appreciate you listening to us for this period, but just some take-home messages. Pam really pointed out the importance of these herbarium collections as powerful resources for these big data analyses of biodiversity. Then what I tried to point out is that these trees of relationships really are important tools. We build those trees of relationships usually using DNA data. I don't think I mentioned that, but that we can also extract the DNA data from the specimens and use that to build these trees. Pam pointed that out. That's a valuable approach, using this metric of phylogenetic diversity for maybe assessing areas that we should be thinking about for conservation. Also, these climate projections that we and others are making that suggest that there are going to be rapid changes in the Florida landscape, the Florida floristic landscape, and then because of association, the animals of Florida as well. So what we're going to see based on these models is an overall decrease in floristic diversity in our state. A lot of species, if they can move quickly enough, will move north. We're going to see more of a homogenization of communities during this period. And I think those are our major messages. I will end with this slide that, it's now possible to use these big data approaches that Pam mentioned, using all of these herbarium specimens and then these big trees of relationships that we also build to look at phylogenetic diversity on really a large scale. And we're actually doing that in the museum with collaborators for the plants of North America. And this is one of our first reconstructions. It's kind of pretty, so we thought we'd share it with you. And what you can see is that the areas of blue have a lot of phylogenetic diversity, red, low phylogenetic diversity. And in some ways, I think this makes sense if you think about it, what we know about North America. The East has a lot of older areas, older lineages occurred here. Think of all the magnolias and things that we have in our Southeast. The West, being a much more recent landscape, has a lot of recently originated groups, and they're actually we have a lot of more closely related species there, and that's why it's red. So we're looking at this in more detail. This involved a tree of 50,000 species and millions of data points. So these are not easy exercises, but they're going to be very valuable as we move forward. And we'll just like to thank a lot of people on the right. We list them here, some we mentioned during the talk, and then our various funding sources, particularly the National Science Foundation, and then also the University of Florida. And so with that, I'll stop here, and Pam will be happy to answer any questions that you might have. And thanks for your attention today. Appreciate it.
All right, thank you, Pam and Doug, for your presentation. That was really amazing. Stacy has been fielding the questions. So Stacy, you want to open your mic and
sure. I had a question about the red maple example that you brought up. Is Aggregators native in the Pacific Northwest, or was it introduced?
That's a great, yeah, thanks for that question. That's a great observation. So one of the things that, well, I'll back up with a little bit of a story. When iDigBio was getting started and all of these digitization efforts were getting underway, a lot of herbaria were very nervous, and not just herbaria, but museums in general were very nervous about sharing all of their data online, because they thought, well, there are probably things that are misidentified. There might be things that have incorrect locality information. these, you know, these museums are incredible sources of information. And in some cases, they have specimens that have been, that were collected 300 or more years ago. But museums are also generally understaffed. And so it's almost impossible to keep up with the new collections that are coming in and at the same time go through all of the backlogs to check the correct label and everything for all of your specimens. But what happened was when people started putting their data online, they got lots of help from the community with regard to noticing outliers in terms of distributions. And so those points that were not in the Eastern US, and you might have noticed that there were some in the Atlantic Ocean, those have errors with them, right? So they could be misidentifications, they could be correctly identified, but when the locality information got switched into GPS coordinates, there might have been an error associated with that. And they could be cultivated. So it is very likely that there are Acer rubrum plants that occur in Seattle, in Spokane, and other parts of the Pacific Northwest. When we were first looking at Acer rubrum as an example for doing this sort of explanation, there weren't any other plants west of the Mississippi really, except for 2 in Colorado. And one was in Denver, looked like it was in Denver, and one looked like it was in Boulder. And we thought, okay, those are probably botanical gardens or cultivated species. So in fact, what we can do is use those locations to go in and understand something about those particular individuals. We can then annotate the specimen records and make notes that say this was cultivated in this botanical garden or something like that. And then they can be excluded. Now, what we do when we actually do analyses, so let's say we wanted to build a model like Doug described and do it for Acer rubrum. Well, we wouldn't want to include those species that are outside of the native range because those could alter our climatic inferences for the species as a whole. So one thing that we do when we do these sorts of data analyses is that we have a checklist for botanical gardens and cultivated areas across North America. There are also botanical gardens that on a global scale that are part of this checking purpose. And you can actually filter out any specimen records that fall within a certain distance of those recorded botanical gardens or known cultivated areas. And so that way we aren't misled by incorporating data from outside of the native range. But of course, you know, there's always a little bit of circularity with knowing exactly what the native range is, because the native range is in large part determined by our understanding of the distributions of specimens. That's how we understand on sort of first principles where specimens are distributed. And so if we then use that to draw a buffer around the points that we use, we could maybe get into some circularity. So we always have to use good judgment in filtering out possible outliers, but certainly by using these lists of botanical gardens, we can get rid of these things that are certainly cultivated plants. I'm sure that's a much longer answer than you had, than you were expecting, but it brings up a lot of really interesting points. So thanks.
Thank you. Is it possible that plant species from areas south of Florida will somehow migrate into Florida to replenish some of that lost biodiversity as a result of climate change?
You can take that one.
Yeah, that's a really good question. And this is something that people are thinking about based on these models. Should we project not where plants are now, but where they might be in the future, and that way try to encompass those areas in some sort of restoration or plan to save those plants? You know, a really interesting story is that timber industries in the Northwest of North America are already doing this. long-lived species like Douglas fir, which is, of course, great economic importance. In Canada, they're already planting it far north of where it now widely occurs because of climate models. They know where it will thrive in the next, or they predict where it will thrive in the next 50, 60 years. So we can do that as well. I think that's a really interesting question, and probably is something that needs to be considered a lot in conservation efforts. Yes, agree.
One other point to maybe add to that, if we think about potentially more tropical things moving into Florida, as some of our Floridian species are potentially moving northward, we see evidence of that in our herbarium specimens and in the monitoring work that organizations like FNA do, because every once in a while, and we can think of some specific examples of some fern species in the Panhandle, for example, that got established, and they're otherwise only known from either the Caribbean or Central America. And yet, botanists will find them periodically, but they don't last for very long. So you can trace back to when it might have been introduced. Maybe some spores were brought in with a hurricane, and the plants are able to establish and persist for some period of time. But when if the weather gets too cold, then they would be wiped out. But if the climate is continuing to warm, it might be that those more tropical elements could move into parts of Florida that are currently, very warm temperate, and could become established there because the climate is different. So some of that diversity, in fact, could replace some of the diversity that's moving northward. But the combinations of species would be very different from the communities that we recognize today. And that could lead to a lot of problems in terms of how those species interact with each other, how they support animals and fungi and microbes and things. So although it's important to have diverse ecosystems, the characteristics of that diversity are really important.
Great, thanks. I have a question about the differences between the plants that you've used in your presentation as examples, why some are projected to thrive not post climate change, but as a result of climate change, whereas others are projected to almost disappear.
Yeah, that's a great question. And it's hard to give a really, totally simple answer because the needs of plants are complex, so in those models, there are soil features, there are precipitation features, there are temperature features. But, one generalization, and you can see this with some of the Lake Wales species, those are fairly dry, hot adapted areas. And so a number of those species, based on these climate projections, where Florida is going to be a much warmer and drier place, based on these climate projections, many more days of extreme heat. Those plants, based on those models, would have a lot more potential habitat. And I think that makes sense when you think about it. In other cases, some plants that are very, cool adapted, which we see a lot of where we are in Gainesville. We're sort of at the southern edge of many temperate elements. That is, those temperature limitations, those cool temperature requirements are going to get pushed further and further north. And so it makes a lot of sense for those species to have to migrate north in order to survive, if they can move that quickly. So some of them are, I think makes sense. Other times we look at things and it's they're sort of, well, we have to scratch our heads a bit. They're a little bit more complicated. But I also need to stress that these models are all based on abiotic factors. We are not able, they're just way too complex to put in biotic factors like pollinators and dispersibility and things like that. So those are all factors as well that make these projections as they are. Just keep in mind that they're just the best we can do at this point in projections. But I think those simplified examples that I gave, they make sense for many of the species and the patterns we're observing. I think if I gave you and we've done this with classes, if I gave you the characteristics of species And I told you based on projections what we think the temperature, number of hot days and drier days will be in Florida in the future. You would make the same projections as the models in most cases.
And that's a good segue into the next question. Regarding the climate models that you're using, is this based on, what is it, emissions that have already happened? Is this future projections based on business as usual emissions, or how are your models determined?
Yeah, that's a really good question too. You guys are all asking wonderful questions. So there is a set of internationally accepted models that are based on the emissions at different rates for the next, let's say, 80 years or so. And some of them are, okay, if we maintain current emissions over the next 80 years or 70 years or whatever the time frame is, then we would expect a certain pattern of temperature increase. If we cut back to emissions levels from 25 years ago, here's the projected climatic condition. If we, if we actually are able to, curb the increase a little bit, what happens? And if we accelerate, what happens? So there are 4 of these general models that that we use. And so we actually do the experiments across all of those different models, and then either average them or sort of take one of the middle ones, which, maybe is the most likely. Hopefully we won't allow emissions to continue unchecked well into the future. But whether we can actually pull them, keep them stable or pull them back is, probably a very difficult task.
Yeah, and I'll just add to that, people often ask, well, their models, how good are the models? Of course they're models. And as Pam said, there are a range of models, and we try a range of models because you can get a range of results, but the generalities we provide are always the same. But I also point out that, really scientists started making climate models and projections probably 40 years ago. And many of those models were pretty spot on. And they were all very spot on in terms of projections for what's happening to the western part of North America with the drying and heat, fires. All of that was projected based on what was considered at that time fairly simple climate models compared to the complex models that we're using today. And this is true for other parts of the world too. So the modelers have actually have done a fairly good job of projecting what things might be now from 30, 40 years ago. So I think these are pretty good landmarks for what we might expect in the future.
So, and we've actually done some analyses of Florida plants that, because again, some of our herbarium collections go back well over a century. So we've done some analyses where we said, okay, let's look at plant distributions before 1960 and then after 1960, and let's see if there is any sort of difference. And in fact, we see already by looking at the subset of the herbarium records for Florida that that since 1960, the average distribution is farther north than before that. So we're already starting to see in this very small subset of data, this move option that some plants at least have, so that they're moving in response to climatic change over the last 50 to 60 years.
Okay, just a couple more questions. Do the models also take into consideration the increase in rain that Florida gets, for example? I mean, they're saying that, the year was hotter, but it was also a much wetter year.
I mean, I'll just say quickly, oftentimes we think of 1 year versus the next. Oh, we got a lot of rain this year. Oh, the next year, not quite as much. So really models are long-term trends. They're not year to year. So you have to think not in terms of what happened this year versus last year or the year, what will happen as a general trend over 30, 40 year periods.
But we can't, but we do actually sometimes build the models based on data from particular years. So I gave that example of the pre and post 1960. We can actually also build into the models the date in which the specimen was collected. And that allows us to match up the date that we know that a species was occurring in a particular location with the actual environmental conditions of that year. But the one other thing that I'll mention with regard to these climate models is that they're large. Okay, so as I mentioned, they're based on emissions, which then have effects on temperature, but those temperature effects control global patterns of precipitation as well. So those future models do take into account the model of precipitation, its distribution through the year, whether it's all in the summer or whether it's more evenly spread throughout the year. So, so all of those sorts of things are accounted for as best as the climate scientists are able to do it at this point in time.
Great. I have one more question. What role do introduced invasive species play in this discussion?
Yeah, great question. You know, so far we've really pretty much concentrated the work that we showed you on what would be considered native plants and maybe naturalized that we know have been here for you know, several hundred years and are part of the flora now. But that's a great question, and that's one of our future projects. What happens with these introductions? And, you know, we can use tree of relationships here. Do they take the place of maybe a native close relative? Can we project how an introduced species might do just based on relationships and other characteristics, so that we could do future projections? So, yeah, that is a hot topic, and that's something that we and a lot of other people are very, very interested in. It's a great question.
Yeah.
Did you want?
Nope,
Okay.
no, great answer. Yeah.
Well, I think that's probably all we have time for. Thank you so much, Pam and Doug, for joining us today, and great information. We do have more questions. If you still want answers to your questions, please email us at info@flaWildflowers.org, and we'll get them to Doug and Pam and get them answered. Thank you again, and we really appreciate it.
Okay, well, thanks a lot.
Thank you. Thanks.
Bye, everyone.
Yeah,
Yep.
bye.
Bye.
Bye.